So let's move to my topic, and it is staining of gel. And one of the most important aspect of gel electrophoresis technique is staining. And one sample molecule has separated in the gel matrix, it is necessary to visualize their position. So this is achieved by staining with an agent appropriate for the sample. So the sample is ethidium bromide for DNA and composite brilliant blue for the proteins. And the components is identified by radiolabeling or by silver staining. So here radiolabeling and silver staining are the methods that we use to identify the components. And gel electrophoresis is usually carried out in the basic pH, and which is most of the biological polymers are anionic hence. So they move towards the anode. Okay. So smaller molecules travel faster than the larger molecules in the gel, and the molecular weight of the protein is determined by running molecular markers in parallel. So in when examining it, gel staining is an important visualization and detection step that follows protein, polyacrylamide and gel electrophoresis such as PAGE and such as SDS PAGE and native PAGE or 2D PAGE. So here we can steady page and HGS page. And composite brilliant blue is a homogeneity dye that binds electrostatically with the amino and carboxyl groups of the protein. Okay, so here the figure of page and its components. So it's having a voltage, and there is in a blocks that we can see, and that's called sample wells. And it have upper buffer tank, and also a polyacrylate gel and electrode, and lower buffer tank. So the page contains two buffer tanks. One of it is upper buffer tank and the second one is lower buffer tank, and it's named because of its positions. And there is some applications for the SDS page, and the applications are to measure molecular weights of the protein, number 1, and number 2, peptide mapping, and number 3, estimation of protein purity, and number 4, determination of protein subunits, and number 5, analysis of the number and sizes of the polypeptide units in a protein.